green plants prepare their own food by photosynthesis. Hence, they are known as autotrophs. Photosynthesis is a physiochemical process in which light is used to prepare food. Hence, it is evident that photosynthesis is the basis of food for all life on Earth, and it is the process that releases oxygen to the Earth. We already know that chlorophyll, light, carbon dioxide and water are necessary for photosynthesis. Let us look at a few experiments that unraveled the secret of photosynthesis. Joseph Priestley, who discovered oxygen, identified the role of air in the growth of green plants. He used a bell jar and a candle in his experiment. He lit the candle and covered it with the bell jar. After some time, he observed that the candle got extinguished. He then performed a similar experiment using a mouse and a candle, and observed that after some time, the mouse suffocated to death. Priestley repeated the experiment, but this time, he also placed a mint plant along with the mouse inside the bell jar. This time he observed that the mouse stayed alive and the candle burned long after he expected it to. So he concluded that the plant was able to replace the air that the mouse and candles had used up. Another scientist, Jan Ingenhousz, used the same setup as Priestley to understand the importance of sunlight to plants. He conducted the same experiment in the dark and in sunlight. He also performed this experiment with aquatic plants in bright sunlight, and noticed that the small bubbles released by the green parts of the plant were oxygen. He demonstrated that only plants can release oxygen. Grana is a membranous structure responsible for trapping sunlight and involved in the production of ATP and NADPH. This is called light reaction. In the stroma, enzymatic reactions incorporating CO2 take place, leading to the production of sugar molecules. This reaction doesn't require light, but depends on the products of light reaction, ATP and NADP, and hence is called dark reaction. Leaves come in many shades of green. This indicates that there are many pigments in a leaf. A chromatographical analysis of green leaves shows that these different colors are due to 4 pigments: chlorophyll A, chlorophyll B, xanthophylls, and carotenoids. Chlorophyll A appears as bright green or blue-green in the chromatogram, whereas chlorophyll B as yellow-green, xanthophylls as yellow, and carotenoids as yellow to yellow-orange. All these pigments are involved in photosynthesis. Each pigment has the capability to absorb light at different wavelengths. If we create a graph with wavelength on the x-axis and absorption on the y-axis, it shows that the capacity to absorb chlorophyll is between 400 and 700 nanometers, which is the visible light spectrum. The action spectrum of photosynthesis shows that maximum photosynthesis occurs in the blue and red regions of the spectrum. We find it is close to the wavelength of chlorophyll A. We can conclude that chlorophyll A is the chief pigment involved in photosynthesis. This shows that the accessory pigments chlorophyll B, carotenoids, and xanthophylls also trap energy, but they transfer it to chlorophyll A. This helps the plant to utilize a wider range of wavelengths from the incident light and also protects chlorophyll A from photooxidation. Julius von Sachs, in 1854, showed that glucose was formed when plants grow. He also found that the green substance chlorophyll is stored in special cells of the plant. He also proved that these green parts are the factories that produce glucose. and it is stored as starch. T.W. Engelmann illuminated green algae cladophora with a spectrum of light using a prism. He placed the cladophora in a suspension of bacteria to pinpoint the sites that produce oxygen. The bacteria accumulated mainly in the areas of blue and red light. The first spectrum of photosynthesis was thus produced. By the middle of the 19th century, the empirical equation for photosynthesis was derived. Cornelius van Niel found that photosynthesis is a light-dependent reaction in which a hydrogen atom reduces carbon dioxide to form carbohydrates. He also inferred that H2O is the hydrogen donor which is oxidized to O2. He came to this conclusion when his experiments with sulfur bacteria released sulfur instead of O2. In that case, H2S was the donor and not water. This was later proved by radioisotopic techniques. This is how the final equation of photosynthesis was derived. Photosynthesis takes place in the chlorophyll pigments of the green leaves and stem. In the mesophyll cells, they align themselves near the walls to obtain maximum sunlight. The chloroplast consists of grana, stroma lamellae, and fluid stroma.